Most living creatures are launched into life from a microscopically small, fertilized egg. And throughout the various stages of growth, a series of complex processes must function perfectly if the final result, be it a giraffe or a tree, a sheep or a flower, a butterfly or a man, is to be recognizably the offspring of its parents. How are exact copies made? Inside each living cell, there has been found a unique molecule which controls production of proteins and replicates itself exactly. And the specific character of each organism is decided chiefly by the types of protein found in each of its cells. Proteins consist of about 20 different kinds of amino acids, combined in a specific predetermined sequence. These small sections of hemoglobin, myoglobin, ribonuclease, and lysozyme illustrate the essential similarity of proteins, whose seemingly small differences, however, are so vital to the life process. This is lysozyme, with 129 amino acids linked together. And this is myoglobin, with 153 amino acids. The three-dimensional shape of each protein, here shown as a diagram in 2 dimensions, is important because it decides how the protein functions. The mechanism for the production of all known proteins involved in the life process of cells is the genetic material stored along the chromosomes inside the nucleus of each cell. Deoxyribonucleic acid, or D.N.A, carries the code for proteins and is the hereditary material of life. It is capable of controlling production of proteins of great complexity and variety. Chemically, DNA is made up of 2 sugar phosphate backbones, linked by 4 so-called bases: adenine, thymine, cytosine, guanine. Adenine can link up chemically with thymine and guanine with cytosine, but not adenine with cytosine or guanine with thymine. It is the sequence or order of these 4 bases which is the code for protein building, and this varies from one part of the DNA molecule to another. This shows only a small fraction of a complete DNA molecule. The potential number of combinations is very large, and this makes it possible for each living organism to have a DNA makeup of its own, and this in turn is the basis of the amazing diversity of life forms. The total DNA in a single fertilized human egg contains around 1,000 million base pairs and weighs about 2/10,000,000,000 of an ounce. When a specific protein is required, the section of the DNA, or gene, which controls that protein, splits apart, exposing the appropriate code. Another molecule, called messenger RNA, then copies the code in every detail. The entire code for lysozyme, an enzyme protein present in nearly all cells, is 387 bases long. When the code copying is completed, the messenger RNA travels from the nucleus into the surrounding cytoplasm where the protein building occurs. The building up of proteins involves several elements present in all cells. Amino acids, here shown as simple geometric forms, enzymes, the catalysts, shown as linear forms, transfer RNA, shown as plug-like forms, used for amino acid transport, and the ribosome, the actual location or site of protein synthesis. It is the ribosome which reads off the code in triplets on the bonding sites. Every 3 consecutive bases on the messenger spell out one specific amino acid. Triplet number 1 is for the amino acid lysine. The correct enzyme recognizes lysine, locates the corresponding transfer molecule, and an energy source enables the two to join together. The transfer RNA for lysine identifies the triplet for lysine on the messenger RNA. Triplet number 2 is for amino acid valine, followed by amino acid phenylalanine, and so on according to the code. The complete lysozyme molecule is made available to the cell. While this was going on with one ribosome, other ribosomes had been following, using the same messenger to produce more lysozyme. The types of protein produced by each cell are characteristic of that cell and its DNA. To ensure heredity, these characteristics are maintained when a cell divides. Each time the chromosomes divide, the DNA inside must replicate or reproduce itself with faultless precision. In this way, the coding process itself is passed on from one cell to another. But sometimes mistakes occur with a code. Radiation, for instance, may bring about the substitution of one base for another. Such a change will be copied each time the DNA replicates itself, and in this way becomes a permanent feature of the molecule. This is called a mutation. For example, if a single base of the DNA code for hemoglobin is substituted, it will result in a modified triplet of bases on the messenger RNA. Consequently, amino acid valine replaces glutamic acid. This relatively small change is responsible for sickle cell anemia in humans. Sickle blood cells carrying modified hemoglobin are different in shape and inefficient in transporting oxygen molecules to the body. When inherited from both parents, sickle cell anemia can be harmful, even lethal, like the great majority of mutations. Occasionally, however, mutations can be considered beneficial. When this happens, animals can be bred to include changes and produce new strains, desirable for man, if not for the cow. Heredity normally is a conservative force, but evolution relies upon heredity to provide for some possibility of change. Without mutations, life would be neither so varied, so colorful, or so adaptable. But it is the DNA molecule with its 4-letter code and its capacity for exact replication which is responsible for the biological identity which normally and overwhelmingly makes a fly, a corn plant, and a man.